to give you a nice little talk about how DNA, the real name of this is how DNA is used to solve 4 murder cases. 2 of them are pretty recent, and 2 of them are old cases. And the old cases, of course, they would have not used DNA to process the evidence in the old cases. So I'll tell you a little bit about myself. I was a teacher for about 50 years. I taught everything from 5th grade to graduate school. I taught forensic science for over 20 years. Now, forensic science is not police science. It's 2 different things. Forensic science is the science that's used to process evidence to present a court case. The word forensics means debate, okay? When I went to high school, we had a forensics club, and it had nothing to do with crime. It was a debate society. So any branch of science can be used in forensic science. Whatever you can apply to the evidence is a science that can be used. So it's chemistry, physics, earth science, entomology, DNA, it's whatever branch of science works, and we're going to talk about some of them. So, I don't know if you remember this, but about, I guess it was the late 80s, early 90s, there was a case on Long Island where a young girl, 13 years old, her name was Kellyanne Tinez. She lived in Valley Stream, and she was savagely murdered, and it was the first, they indicted her neighbor, whose name was Michael Golub, and they brought him to trial and eventually convicted him. And it was the first case on Long Island where DNA evidence was used in the trial. The jury did not really use the DNA evidence to finally come to the conclusion that he committed the crime, which is interesting. I watched the whole trial. The prosecution brought in expert witnesses to explain the DNA, because what happened in the crime was that she was stabbed several times. There was a lot of blood. And when somebody uses a knife or a bayonet or anything to stab somebody, oftentimes they'll cut themselves. And so their blood will be at the crime scene. When there's a violent crime, I used to tell my forensic science students, the next time your mother is defrosting a chicken, when the chicken is defrosted, go over and stab it, okay? But don't do it in front of the family, and don't tell them that I told you to do this, okay? Because it takes a lot of effort to penetrate flesh and bone. So when you're stabbing somebody, you're using a lot of force. They bleed, your hand gets slippery, and you slide down the knife, and you cut yourself. So your blood can be at the crime scene. So it was, and so what they did in the Kellyanne Tinez case was it was on her clothing. What he did with her was, after he killed her, he put her in a sleeping bag sack and stuffed her in the closet. And what happened was she got a phone call from the Golub residence she's 13 years old, to come because Michael Golub's little brother was in the same class as she was, and so they suspected that she was lured there for a homework assignment or something. She was minding her little brother. So she went, and then the parents came home from work, and she still wasn't back, and they said, where was she? And they said, the Golub, she went down to the Golub. He said she went down to the Golub house. So the family waited until 11 o'clock, and then they called the police, and the police came in, and of course they figured she was a runaway. You can't file a missing persons report for 48 hours. But the next morning, she still wasn't back, and the police went to the house just to check. They went to the Golub house. And the mother opened the door. She had no idea what was going on. And the policeman said, do you mind if we just look around? And she said, sure, fine. They went into the basement, and they found her in the closet. So what they did, what the police must do at that time, is they must stop doing anything. And they must secure the crime scene. They need to get a patrolman to come and secure the house. And then they have to go get a search warrant. Okay, Okay, so they got the search warrant and they processed the evidence. And what happened in the trial was that they had expert witnesses and they went on and on and on about this DNA evidence, and nobody knew anything about it. So the jury was a little bit confused, I guess. And so what they did at the end was he bit her. In the process of killing her, it was so long, he bit her, and he bit her before she was dead, so she bruised. So she had bite marks, okay? So the bite marks were, what they did was they took a cast, like a dental cast, of Golub's teeth, and they matched the bite marks. But the defense also had an expert witness who said, no, it doesn't match, look, and he was really off one tooth. And so the jury, again being confused, what they said was, bring us the pictures of the bite marks and bring us the cast of the teeth. And what they did was they took the arm of one of the jurors and pressed the cast into their arm and then checked it, and that's what they convicted on. So it wasn't the DNA, even though it was the first case on Long Island using DNA evidence. Okay, so what I'm going to talk about today, as I said, are these. You have to wait till my eyes adjust to the dark here. Yeah. These four ladies. Okay, so I'm sure that you recognize the lady on the right. That's Nicole Brown. Her picture was in the paper often. The lady at the top, that's Lacey Peterson. Lacey Peterson was 9 months pregnant when her husband killed her and the baby. Okay, the lady on the left, I'm sure you don't recognize, her name is Marilyn Shepard. Did you watch The Fugitive?
Yes.
Okay. The Fugitive was a TV show that was based on the Marilyn Shepard murder case, and it was very fictitious. Okay, the lady on the bottom, her name is Mary Sullivan, and I'm sure no one recognizes her. Okay, so we're going to talk about these four cases. All right, let's go here. Okay, so that's just what I said. Okay, so these are some of the factors that will affect a case. Storage of evidence is very important. I'm going to be talking about two old cases. Well, they go back to the Mary Sullivan goes back to the early 60s, and Marilyn Shepard goes back to the 50s. So the police keep evidence for almost forever, which is very good for a number of reasons. So they had the evidence to then come back and reapply DNA technology to the evidence in the old cases. And of course, we know in the O.J. Simpson case, I'm sure you were very aware of watching this thing almost every day. And there was a mountain of blood evidence in this case, and there was all this blood evidence was processed using DNA technology. And what happened really was that the defense team tore apart the methodology and the expert witnesses of the prosecution. And so doubt in the mind of the jury, not only for the evidence, the way it was handled, but also of the detective who handled the case. His name was Mark Furman, and what they did was they found out that he had used the N-word on a number of occasions, and when he was interviewed, because O.J. Simpson was an African American, they asked him, did you use that word? And he said no, and then they got a witness who said he did. So what they did was they impugned his honesty. So if he was dishonest about that, was he dishonest about he was in charge of the case. Was he dishonest about the collection of evidence? So that doubt was sown in the minds of the jury, and I think it played heavily in their decision. Okay, so the acceptability of DNA evidence. Let me just see if I have something on the front. No, I don't. Okay, so, okay, we're just not going to go back. Or maybe here. No. Okay, so acceptability of evidence is ruled by court decisions. In other words, you just can't come into the court and say, all right, I did this test on this piece of evidence and this is what I got out of it. Any scientific method that is used today comes under two decisions, the Fry decision and the Daubert decision. The Fry decision very simply says, if the scientific community accepts the science, in other words, they say this is a valid test and it passes their scrutiny, then the court will accept it as well. The Daubert case goes a little bit farther simply because scientific evidence has become such a big part of criminal and civil cases. At one time, the courts depended upon eyewitness accounts, confessions. They really didn't use science all that much. Now, prosecutors and defense attorneys, they want to see hard evidence. Okay, what is evidence? Evidence is any item that can connect the criminal to the crime scene to the victim. Okay, so and when you think about it, when the crime scene unit goes to collect the evidence, how do they know what to collect? They know what to collect because they know what the crime lab is capable of processing. So the most obvious one is fingerprints. Okay, so fingerprints have been considered for years and years to be the best way to identify somebody, and now that's even coming under a little bit of scrutiny. So you know that when you inherit your fingerprints from your parents, you inherit the patterns. They're called loops, whorls, and arches. Well, you'll inherit that, but then there are always variations that occur. Even in identical twins, that's always the question, do identical twins have the same fingerprints? No, because during the gestation period, before they're born, they touch things. They touch the inside of the uterus, they touch the amnion, they touch things and they get accidental marks which make their fingerprints different. And then the minute you're born, you start touching things. And you know yourselves, you have scars on your fingers, you change your fingerprints as time goes by. So, all right, what else is, the next thing that we need to think about is, again, the law. Evidence cannot be collected without a search warrant. Okay, and that's good. That's an excellent thing. So what I'm really going to talk about here is DNA technology, and I don't want you to go to sleep, okay? But I'm going to tell you how it works. I'm going to tell you how DNA works. Okay. This is a chromosome. We have 23 pairs of them. How many of you have done Ancestry? Yeah, isn't that great? I did it and I thought to myself, my ancestors come from Poland, but I thought, the French went through Poland with Napoleon, and the Germans and the Russians. Everybody tramped through Poland at one time or another. So I thought, gee, what have I got in my genes? So I did it and I'm Polish. What can I say? Okay, so the chromosomes are located inside your cells, and there's a structure inside the cell called the nucleus, and the chromosomes stay in there, okay? They do not leave. But at some time, during what is called a resting stage, a cell never rests. If the cell rests, you're dead. Okay, so during what's called a resting stage, the chromosome begins to unravel, and it's like a slinky. Okay, a slinky when it comes in the package is really tight and then you can pull it out. Well, that's what happens with DNA. And in really, if you just kept pulling it and pulling it, it would eventually become a ladder. And the rungs of the ladder, if you look at that diagram, that's where the code is. Okay, we know that DNA is called the code of life. And what it does is it instructs the cell to produce the substances that keep you alive. Okay, a chromosome, this is something that's probably more familiar to you. Chromosome is made of discrete little pieces called genes, right? And the genes determine your characteristics. But what the job of one gene is to make one protein That's all it does. One gene makes one protein. That protein can either be structural, because we're made of protein, or it can be regulatory. It's either a hormone or an enzyme. Okay, those of you who take probiotics, you know that the probiotics are all enzymes. And enzymes are substances that control chemical reactions. So this is why your genes are important, because the genes will translate into body chemistry. And body chemistry is what you are. The color of your eyes, the color of your hair, how tall you are, whether you have freckles, whether your ears stick out or they're close to your head, all of that is controlled by genes. Okay, so how is this used in criminology? Okay, we're not even going to do that because I just said that. Okay, so this is just a nice little diagram that shows that what happens with, this is how this works. This is the nucleus of the cell, and this is where the DNA is. So when the DNA, remember I said when the cell is resting, the DNA is all unraveled? Well, during that time, the DNA makes RNA. Everybody knows what RNA is, right? Why?
No.
You don't know what did you get a COVID shot? It was an RNA vaccine. In other words, the vaccine was produced from an RNA molecule that had instructions in it that would make a protein that would latch on to the virus and prevent it from causing a disease. So this is a whole, you know, your normal vaccinations are simply like a flu shot is the killed virus. And the killed virus has proteins, and the proteins, when they enter your body, cause you to make antibodies, and so then you don't get the flu, you hope. Okay, so anyway, when the DNA is resting, it's making half a ladder, okay, called RNA. The RNA travels over here to these little bumps on there, and they're called ribosomes. That's where the proteins are manufactured. But let's move on. So there's a lot of technologies that we have now about DNA. We know so much more in the last 10 years than we knew in the last 2000, okay? So it's an incredible technology. This is DNA sequencing. What does that mean? That means that we can unravel the code. The code comes in 4 letters. They're not really letters. In other words, your cell is an alphabet soup. Okay, the letters stand for small molecules. Like the A is an adenine molecule, and they hook together those 2 molecules to make the rung of that ladder. But we can sequence it. What does that mean, sequencing DNA? Okay, let's suppose we have an endangered species. Okay, take their DNA and sequence it and save it, and possibly we can, if that species becomes extinct, reproduce it. Okay, here we go. So this is the way it is. The DNA makes up chromosomes. Chromosomes are made up of genes, and the genes come in various variations. Okay, I'm going to talk a little bit about the tools that are used in forensics, okay? We have something called restriction enzymes. A restriction enzyme is able to cut into a DNA molecule. This is important in forensics because what we do in forensics to process evidence is we need to cut certain little pieces of DNA out of the evidence. In other words, I can make a profile of you by cutting out only certain little segments. We have a lot of DNA. Most of our DNA, we don't even know what it does. We used to call it nonsense DNA, but we're better now. We don't call anything in the cell nonsense. Okay, so, but what is important are tiny little pieces. How do we know about those little pieces? Basically through medical research, because in medical research, doctors realized, or microbiologists realized, that diseases are caused by genetics. Many are caused by your genes. And what they tried to do was develop a test where they could look at your DNA and see if there was a little marker in there, a little piece of DNA that said, aha, you're going to have that disease. And they were very successful at that. And you're going to see that that's actually played an important part in criminology. So restriction enzymes can cut the DNA and cut it in specific places. So in other words, I can target a piece of DNA that I know is a marker and pull it out. Okay? I don't know if you've ever heard of CRISPR. CRISPR is a DNA tool now which enables people to manipulate DNA by cutting it in pieces, so you can open up a chromosome and insert a gene. If you had a faulty gene for a particular trait, you could take the right gene and insert it. And that's a technology that's already used. Okay.
Excuse me. If you insert the gene, does that get passed on from that person, or?
Only genes only get passed on from sex cells. So if you inserted them in sperm cells or egg cells, or in the part of the ovary that produces the egg cell, or the part of the testicles that produce the sperm cell, if you insert it there, yes, it'll be passed on. If you do it in the body cells, no. Okay?
Thanks.
Oh, you're welcome. All right, so all of these terms up here, RFLP and PC, it is alphabet soup. This whole DNA technology is alphabet soup. These are two techniques that are used in criminology. RFLP is going through your DNA and only cutting out specific markers and making your specific pattern. So think about that. If I have a suspect and the crime left blood at the crime scene, I take the blood of the suspect, I run this RFLP, in other words, I cut out all these little chunks and I make his pattern. I make his DNA profile. Then I take the evidence and I do the same thing. I put them side by side. Do they match? Okay? And scientists are so careful, even though scientists sometimes have a bad rep, they are so careful, they will never say it's 100%. They will never say, we lay people say match, but they don't. They say a concordance, and they say it's 99.9%. Okay, I had a student when I was teaching forensic science, and she brought in a DNA report from a laboratory, because what happened was when she was ready to go to college, her mother and father were divorced. And her mother said, I'd like you to help to pay for the college education. And he said, she's not my kid. So she went to court, and she got a court order, and he had to submit his blood, probably spit. And they did the DNA, and it was, I saw it, they blacked out the names and whatnot, but it was 99.9%. So I guess the court ordered him to pay some, something anyway. PCR, if you had a COVID test and you had the PCR test, okay, so the way that works is you cut out little chunks of DNA that are specific to the COVID virus, and the PCR machine actually multiplies them. It's like a Xerox machine. And then you can test exactly whether that protein is there or not. In other words, it will only duplicate the protein that they're looking for from the COVID virus. And if it's not there, the test is obviously negative. Electrophoresis is just a technique. You see those blotches? That's how you make the profile. That's how you separate those little chunks that you cut out and make them visual. Why? Because the little chunks have a negative charge, and you sit them in this little tray and you subject it to an electric current, and they migrate. And they make and you will see what it looks like, because we will go to this gentleman who is actually probably responsible for using DNA technology initially. His name was Alec Jeffreys. He's British. He was working at the University of Leicester, and he was doing research on disease. He was looking for those little DNA chunks that would indicate that a person had a specific disease. And what happened was there was a case in the newspaper of a Ghanaian family, and there was a young man who was staying with the family, and the British version of Immigration and Naturalization Services said they were going to deport him because he really didn't belong to that family, that he was lying about that. So Alec Jeffries reads this and he says, I can prove whether or not he belongs to this family. And what he does is he only has certain chunks that he's working with. It isn't that, these scientists have every little chunk of DNA. He only had certain ones, but they were enough. So he said, I will take the young man's DNA and the family's DNA. I think he was staying with an aunt or an uncle, and we'll see if they match. And so he proved that he did belong to that family. So he saved that kid from going back to Ghana. Okay, so what happens in Alec Jeffries' life and in England, actually this happened in Yorkshire, was there was a serial killer. And he was killing young ladies, and they were going nuts. I mean, everybody was, if there's a serial killer in your neighborhood and people are dying off, you're really very nervous about it. So Alec Jeffries contacts Scotland Yard, and he says, if you can get all of the men in this neighborhood to give me a blood sample, I can, and you let me, have access to the evidence, I can find your killer for you the same way, okay? So they say yes. So they have all the men in that particular, I guess all of Yorkshire, submit a blood test. All right. The killer paid someone to take the blood test for him. He changed his identification card, and he got this other person to take the test for him. The other person was not the sharpest pencil because he goes to a bar. The guy who takes the test for him, he goes to the local pub, and he's with his mates and whatnot, and drinking his ale, and he's saying, you know what I did? You know what? And the murderer's name was Colin Pitchfork. How do you like that? Colin Pitchfork. So Colin Pitchfork, he said, maybe Colin, he paid me £200 to take the test for him. And his friends are looking at him like, what? And you didn't figure this out? They said, you have to go to the police. And sure enough, they arrested Colin. They did the old DNA test, and it was Colin. So, very interesting, Alec Jeffries. He's one of my favorite characters. Okay. All right. So that's what, I showed you that electrophoresis where the electric charge is used to separate the chunks. Well, there they are. That's what it's called, an autorad. Autorad. And it's like a piece of cellophane tape, and it has lanes on it. And I'm going to show you that in a minute, in a very simplified form. And you can see it pretty much in this picture that you can make matches or not matches. Okay. So, all right, now what I'm going to explain here is what the difference is between people, okay? Because when you're doing DNA technology, you're really not looking, this technology, you're not looking at the gene, because the gene's going to be the same. Our genes for insulin are pretty much the same. But what scientists discovered was that the genes are surrounded by little traffic signals. And I'm showing them as X's, okay? And what they're finding is that little run, it's really like a run of DNA letters that repeat themselves. And they repeat themselves before the gene and after the gene. So what's the difference between A, B, and C? The length, right? It's the length of that run. So that's where your RFLP comes from, because it says restriction, fragment, length, polymorphism. In other words, what it's doing is it's cutting these pieces out. Not the gene, the heck with the gene. Cutting these pieces out and comparing them. Okay? So if my killer has this pattern, but the evidence has this pattern, he's not my killer. Okay? Because that's what we're looking at. Just think of it as scissors cutting a long, that candy where there were dots all over the paper? Just think of it as that, and then there are only certain chunks that you want. And you're always looking at the specific chunks and comparing this person's chunks to that person's chunks. That's really basically what it is. Okay, so And that's what I just said. So let's do this. See, this is a simple one, okay? Your auto rad will have lanes like this, okay? And what we have here is we have a paternity suit, all right? We have mom and dad, and of course it's so obvious because of the colors, but then we have all of these children. We have 2 daughters and 2 sons. Well, which ones of those children, those 4 children, belong to mom and dad? Well, because they color-coded it, it's pretty easy, right? But you can see how it works. I mean, this daughter 1, has this gene from her mom, and this one from her dad, and this one from her dad, and this one from her mom. So obviously, okay, if it wasn't color-coded, it would be a little bit more difficult. And then, of course, so is this one. But the way you match it is, and that's the hard part of this whole technology, is to make sure that your tests are run carefully, because if you don't run them with exactly the same the same guidelines, those little pieces will be farther apart in one than they are in another, and you'll never be able to. I took a course when I taught forensics. I took a course at Stony Brook on doing this stuff. I found it very difficult, and I mean, I taught chemistry for years, but to be precise was very difficult. So,
excuse me, could I ask a question? The green guy,
yeah.
the beanie cat. Okay, he's pure. The
Who
one
knows
to the
where
right,
he came from?
with the right doesn't match. There's no yellow or blue. And I'm sorry, the pigtail girl and the one on the end. In other words, neither one of them were parents to those 2 children at all. It wasn't that mom was a floozy or
Well,
got raped or something.
Mom might have been.
Wouldn't there be some trace?
Yeah.
Mom?
See, mom has
Yeah.
this one and this one in this kid.
Oh, okay.
Okay.
Yeah,
I'm sorry,
I
but
see.
what about this one?
There's
There's
no
this. Okay. And then there's this one and this one. So obviously mom was fooling around. She must have been. I mean, because those blue pieces come from mom, and maybe she was married before.
What about the kid on the end then?
No, who knows? I don't know.
This dog left him on the doorstep.
Yeah. Yeah. Something. Okay. And all right. So PCR. What's the significance of PCR in criminology? PCR is a process whereby you can select a chunk of DNA and duplicate it millions and millions of times. So if I have a teeny little drop of blood with a teeny little bit of DNA in it, I can use PCR and make lots of DNA, and it makes it much easier to compare. Does that make sense? I hope so. Okay. So, all right. So, I have to mention this because this is used a lot in forensic science. There's only certain, again, what I tried to point out to you is that they're only using certain little pieces of DNA. Well, we find that the pieces of DNA that come from the X and Y chromosome are very helpful. And you're going to see in one case it's been very helpful. The XY chromosome pair is what determines your sex. If you have 2 X's, you're a female. If you have an X and a Y, you're a male. Okay? Women have more DNA than men do. See? Men are missing one of those legs. I don't know what that means. Well, with chromosomes, more is not always good. Okay, for instance, Down syndrome kids have an extra chromosome. So that's not good. Okay, so let's talk about the Sam Shepard case. 1955 to 1956. What happened here? Okay. Well, Sam Shepard was a doctor in a small town in Ohio, a very affluent town, and he was married to Marilyn, and Marilyn was very attractive, and so was Sam, and they were both very socially active, and they were both cheating on each other. And he came home one night from whatever, and he found her murdered in her bed very violently. with sexual overtones, really terrible. But he was a doctor, so what was the first thing he was going to do? He was trying to see if she was still alive, if he could help her. So he leaned over the bed, and he got his hands all bloody, and he, for whatever reason, took off his shirt. He took, I think, about an hour or so to call the police. And what he claimed, and this was in The Fugitive, that there was a bushy-haired man who came in. When he came into the house, the bushy-haired man was there and hit him. So he was knocked out for a short period of time, and then he found Marilyn and tried to revive her, and then called the police. And the police came, and they were very suspicious of him. I never knew why they were suspicious of him, but they were, and they went after him. And eventually he had to stand trial, and let's see, and we know that that was the basis of The Fugitive. Okay, so this is just what I said. Sam Shepard died in 1970. This is a very interesting case in what happened to him. But he was convicted in 1954, and then he was retried, and he was exonerated in 1966. But by this time, his career was ruined. He became a wrestler, and he died in 1970. In 1996, when DNA technology became acceptable and useful, his son had his body exhumed and compared to the evidence, and the evidence was pretty much degraded. I'm going to talk about the evidence, but the evidence was degraded. But so the lab technician who did the DNA test said that it was very hard to determine without a shadow of a doubt that there was that Sam was innocent, but what he said was that it pointed to his innocence, and I'll show you why here in a minute. Okay, the evidence to convict Sam was his alleged confession. Let me tell you what the confession was. He wrote a book, and he signed it to a friend, and in his signature he wrote, "I did it, Sam Shepard," and the "I did it" was, "I wrote the book." Now, I've written books, and I can understand saying, "Wow, I did that." So, okay, they had a dog that didn't bark. So what did that indicate? It indicated that the dog knew the person in the house. That doesn't mean that the person was Sam. This is all circumstantial. There was no forced entry. That means somebody had a key, or they knew how to jimmy a lock without it showing. Sam took his shirt off and it was missing. They never found it. And the prosecution said it was because it was full of her blood. Okay, but they never found it. He delayed in reporting the crime. The crime was staged, and see, that really worked against the prosecution's case, because even though they cheated on each other, they weren't mad at each other. They had an amicable relationship. It was known throughout the community that they got along, so why would he kill her? Also, there was damage in the house he had trophies that he won for various sports, and they were smashed. So, would he smash his own trophies? So this made no sense. I just got the feeling when I researched this that they were out to get him for whatever reason, and I don't know why. It was a crime of passion. There were 27 blows, but if they were getting along, why would he do that? There was infidelity. There was a thumbprint on the headboard. Well, because he touched her. He tried to see if she was alive. It was so silly. There was a missing table lamp. They thought that the table lamp was the weapon because she was bashed in the head as well. And there was no type A. And I don't know why I put that there, because that doesn't belong there. So pretend that isn't there. Okay. Now, this is what happened. There was a handyman. His name was Richard Eberling. He had done work for the Shepards. He had access to the house. The dog knew him, okay? He had made a pass at Marilyn. She rejected him. He murdered another of his customers, an old lady, and went to jail. And in jail, he confessed to the murder of Marilyn Shepard. He had her cocktail ring, okay? So it was actually Richard Everling who committed this crime. And when they finally did the DNA evidence, and this is what I never understood about this. The scientists who did the DNA evidence found 3 different blood types. Marilyn's, Sam's, and another unknown blood type. Now, the blood was, you can imagine, she was hit with this lamp violently and stabbed, so the blood was spattered all over the place, plus the fact that there was a trail of blood leading out of the house, which was obviously made by the murderer. He stepped in the puddles of blood and he walked out of the house. And so they were able to get those, they didn't get it from the house, but that had been collected previously, and he found that there were 3 blood types, and the 3rd blood type matched Richard Everling. So, there we go. And then when they exhumed Sam Shepard's body, of course, they would have had records also of his blood type. He did not match all the evidence. So there he is. So there it was. This is the report. Mr. Gilbert said the new DNA testing also revealed that a recently rediscovered sample of blood taken from a large spot on a closet door in the bedroom, only a foot from where Mrs. Shepard was killed, did not match either Dr. or Mrs. Shepard. Is there a blood type there? But it's not rocket science. And it too pointed to Mr. Everling. Okay, Richard Everling cannot be excluded as a suspect, Mr. Gilbert said, using the precise language of DNA analysis. Everling shows up everywhere with his DNA on the closet wall, in the bedroom, on the blood trail on the stairs and on the porch. So, and on Dr. Shepard's trousers. So he must have brushed up against some of the blood. That was, now why did Richard Everling bleed? Because he stabbed her and he used a knife. And as I showed before, that's what happens. This is the evidence used to acquit Sam Shepard. He was injured. He claimed that he was attacked by the bushy-haired intruder, and he had injuries. And they were injuries that he couldn't inflict on himself. The couple's recent affectionate behavior, there was no record of violent tendencies in Sam, the damaged trophies, the lack of significant blood on Sam's pants, socks, and shoes, sperm from Marilyn's, because she was raped also, it was not Sam's. So when they finally did DNA technology, they would have had to exhume Richard Everling's body, or they would have had to have evidence from a previous crime to prove that the sperm was his as well. Closet blood stain matched handyman Everling, a window washer used by the Shepards, Sam's lack of motive, Sam's lame story, no confession from Sam, Everling's possession of Marilyn's ring, and familiarity with house and jailhouse confession. Well, what do you want? Okay, so there is a case where the man's life was completely ruined, and later on, when they had the technology available, he was proven to be innocent. And what I never understood was, how did the initial crime scene miss the blood that belonged to Richard Everling? They just said it. I just read the report. It was all over the place. And I suspect that somebody didn't like Sam Shepard. Okay, so that's what we just said. Okay.
Could they have hidden that evidence?
I'm sorry?
Would the police maybe have hidden that part? The blood from Everly?
Yeah, they would have
If
had to.
someone's out to get Sam and you have the other blood, you just don't present it?
Could be. That's a theory. Okay, next one. This one we all remember, I think. I was teaching forensics when this was going on. I actually used it as my final examination. And what happened here was they were married. She was 9 months pregnant. It was Christmas Eve. The last they saw of her was she took the dog out for a walk and then she disappeared. And Scott's alibi was that he went fishing. Okay? And so when there's a murder committed or a violent crime like this, the first person they look at is the person closest to you, because you have the most reason to do it, right? Unless it's a serial killer or something like that. But that's who. And they looked at him and they questioned him and they searched the house. They couldn't find anything in the house, no blood. Now, there's a way to find blood. If somebody bleeds on your floor, more than likely the blood is going to seep into little cracks and crevices and you're not going to be able to scrub it out. Okay, so they have this stuff that they spray on it. It's called luminol. And it reacts with your blood and it glows. So they spray it, turn the lights off, and spray it, and it glows for a short period of time. And what it does, it reacts with the iron atoms in your blood, and it changes the energy level in the iron, and it emits light. They couldn't find anything. They went into his garage, and they found rope and bags of cement, and this just made them curious. He had a boat. Obviously, he went fishing, so they searched the boat. They couldn't find any blood. They found a single hair of hers on his boat. Doesn't mean anything. They were married. She could have been on the boat at any time, correct? Okay. So they had to start with that, and what they did was they tested the hair, and they determined, they used what was called mitochondrial DNA. Mitochondrial DNA is DNA that is inherited through the maternal line. Everybody's got a mother, so everybody gets mitochondrial DNA. Mitochondrial DNA is not found in the nucleus of the cell, it's found in a little structure outside called the mitochondria. So doing the mitochondrial DNA, they found out that it was consistent and that was her hair in his boat. Okay, so he had a girlfriend. He had very strange behavior. He dyed his hair, when they picked him up, I think he was on his way to Mexico, but he had a girlfriend, and she agreed to wear a wire, and was hoping, the prosecution was hoping that he would confess to the crime, but he came very close, but he never did. So all right, he goes to trial, and I'm thinking to myself when I'm watching this, they have no hard evidence, and juries like to have evidence. They want to have something to go on, not just the fact that he seems like an SOB. Okay, so what did they do? Okay, the prosecuting attorney, what he did was he said, okay, he went fishing. He had a boat. He had every opportunity to take Lacey on the boat and dump her in the water. Okay, logical. All right. So they also confiscated Scott's computer, and on Scott's computer, what they saw was he was looking at the tide and current charts in San Francisco Bay at the place where he was fishing. He told them where he went fishing. Okay. He was so confident that he was going to get away with this, and he was looking at the tide and current charts because he wanted to make sure that the body would wash away. Okay, so the prosecuting attorney said, I am going to look at the tide and current charts too, and I'm going to find out how long and where the body would wash up if he dumped it in the water. Logical? Makes sense. Okay, so he does, and lo and behold, in the allotted time for the body to wash up, remains wash up on the shore at the specific place that the tide and current charts said they would. They were unrecognizable, no head, no hands. I doubt very much if Scott cut them off. I think what he did was he wrapped wire around her neck with cement weights, and when she was in the water long enough that the wire cut her head off, God knows where her head went, and her hands as well. But it was a torso, and then they found the remains of a fetus, a very well-developed fetus. So they collected the remains, and what they had to prove was that the remains were Lacey and Connor. Connor was the baby's name. And so what they did was DNA testing. And they took DNA from Lacey's mother and father and proved that the torso was Lacey, just like that, that paternity suit I showed you. So, and then they had to take DNA from Scott and from Lacey to make sure that the fetal remains, so the baby was like ready to be born. The baby's remains belonged to Scott, and they did. So they proved using DNA technology that that's who this was. And so this was the evidence that they used in court, and this convinced the jury. I never thought it would happen. I thought, that they'll come up with something else, but there's a summary of the evidence. Lacey's hair on Scott's pliers. Oh, yikes. So he used the pliers probably to tighten the wire around her neck. And he had a large amount of cleaning supplies, which he probably knew because they didn't find anything in the house. He must have known some cleaning supply that would really clean up whatever was in the truck, the boat, the house. Scott's boat and fishing habits, his extramarital affairs, Scott's computer search of time and current charts, Scott's strange behavior. He took a lot of money out of the bank, and the location and the identification of the remains. Okay, no tissue from Lacey found on Scott or his possessions. See, this is what, but I was glad the jury found him guilty because I'm pretty sure he was. So, all right. The branch of science that's used to determine what happened to remains that are found. Remains are found like, let's take something local, the Gilgo Beach murders. I live about 3 blocks away from the serial killer, by the way. Lucky me. But anyway, they found the remains in Gilgo Beach, right? Okay, so they use a branch. Taphonomy actually is every branch of science that you could use to try to figure out how that person died. What were the circumstances of their death? You can use bugs, because on a body, any organic material dies, the first thing that moves in is the old flies. And then they do their work, and then make the organic material, we'll call it, hospitable for the next species of insects. And there's like an ecological succession, and each one of them takes a specific time. So by the insect carcasses that you see around the body, you can tell how long the body's been dead. Now, if it's end stage, then usually there's nothing but bone, so you can't really tell the age from that. But all of these things, bacteria, plant material that's found on the body, all of these things can be used to tell where the body originated from, how it traveled from one place to another, if in fact it did. And that's really the branch of science that was used for the remains of Scott and Lacey. Okay. Well, we already said this. Okay, yes, isn't this lovely? Women write to Scott Peterson in jail. Want to get to know him better. Okay, Nicole Brown and what can I say? This was such a travesty. This was hard to watch, for me anyway. And what happened again was we had a mountain of evidence. Okay, you can see OJ there. Because OJ got his comeuppance anyway, because they got him on the civil trial, and then he tried to steal his trophy back, and he kidnapped somebody in Las Vegas. And kidnapping is a federal crime. So they eventually got him, but I think he's out now. And you can see the blood on that grating. Okay, so here are some photographs, and you can see footprints. This is the walkway from Nicole's residence. She lived in a condo, and then he lived in a mansion on the other side of town. So, but they visited both residences. Actually, the police went to OJ's residence because they saw the violence of the crime, Ron Goldman and Nicole Brown, and they were concerned for O.J. Simpson. They thought that this was some sort of a plot, maybe, and so they went to his home to make sure he was okay. And when they got there, of course, they saw him in whatever state he was in, and they started to search his house. Hopefully, they did get a search warrant, and they found blood in the Bronco, and they found blood in his bathroom. They found it in the drains and on a pair of socks. And this was also a very violent crime. So there was a lot of blood. Well, what you see here, okay, is that's the auto rad, and I can't really, it's really very hard. I should have really turned it, and you see the writing on the right, those are the lanes of the various blood stains and evidence, and so they would be able to match it up. And okay, so the summary of the evidence. Well, this was all the evidence, but it didn't matter. Simpson's blood was found in or on his Bronco, his driveway and foyer, the bloody glove, the bloody socks. DNA analysis linked Simpson to the murders by its presence on Nicole's walkway, his driveway, the Bronco. Nicole's blood was on his socks. Ron and Nicole's blood was found on the bloody glove. Remember the bloody glove? Was that the silliest thing you ever saw? First of all, the person during the trial who was putting it on was trying to put a suede glove that was soaked and dried over a latex glove.
Yeah.
First of all, if you soak a leather glove and it dries, it shrinks. Okay, and then did you ever try to get something over a latex glove? So that really annoyed me. But that's okay. The interesting thing here was the socks. The defense claimed that the crime scene people and the detectives in charge actually collected blood in a tube from Nicole's residence and poured it on OJ's socks when they went to his home, and that's how the blood was found on his socks. And that's all you saw on the headlines. But what happened was the prosecution said, okay, if that's true, the blood that was collected in the vials from Nicole's residence would have had chemicals added to it to keep it from coagulating and decomposing, because you don't want the proteins in the blood to decompose because you can't type it then, nor can you do a DNA on it. So the blood, if they were right about that, if the defense was right, the blood on the socks should have had the chemicals in it. So they took it and they did a very simple chemicals test called spectroscopy, and they found that there were no chemicals. It was her blood. So it was frustrating. Okay, so no weapon or bloody clothing was ever found. They think that he was driving to the airport. They think that he just dumped it in a receptacle somewhere. The slow speed chase, of course, we all remember that. And the claims of incompetent police corruption. Yeah, this was really a shame because that's what they did. They said that the crime scene unit was the crime scene unit personnel were not well trained, that they didn't follow protocol in collecting the evidence, and they had the foremost American blood spatter expert, and the defense attorney confused him to the point where he didn't know what he was talking about, which was surprising to me. And then Judge Ito just did not really have a tight courtroom. So with all of those things, and basically what I said before is really what it was, is that they actually impugned the honesty of the lead detective, and that figured heavily in the jury's lack of conviction. Okay, Mary Sullivan, her crime was not solved in 1964. She came from Boston. She got a little apartment she came from a small town in Massachusetts. Came into Boston to get a job. She had a job. She was just starting a new life, and she was murdered. Of course, they saved the evidence, and what happened was eventually they were able to connect the evidence from this case, from Mary Sullivan's case, to Albert DeSalvo. Albert DeSalvo, they didn't know who the Boston Strangler was all the time, but it eventually ended up as Albert DeSalvo. And he was actually murdered in jail. He was convicted of another murder, put in jail, and he was murdered in jail. And he was obviously buried, but he had relatives, which is a good thing. And what happened was, once DNA technology became commonly used, they reopened the case. And what they did was, the first thing they did was they took a DNA sample from his nephew, whose name was Anthony. And they found that Anthony's DNA was not a complete match, but was a close enough match to the DNA in the evidence. They had sperm evidence from her murder. There was enough of a correlation that they were able to get a court order, and they exhumed Albert DeSalvo's body. And they took DNA from his teeth and from his bones, and it was a match. Okay? And they used something, this is a brand new technique. They keep improving this technique, and you'll see it if you follow different cases. It's called YSTR. And Y is because it's on the Y chromosome. And that was why it was so important that they had to get a male relative of his, because it's only the Y chromosome. And the STRs are very tiny, so they're very easy to excise from the DNA and match. And that's something that they use often. Even hair. When I first started teaching forensic science, hair would never be considered to be what is called individual evidence. Individual evidence means that you can connect it to one individual and one individual only. Whereas class evidence, like if I test blood and I get type O, that's half the population. That's class evidence. So now, as I was saying, hair was because unless you get a root from the hair, there's no DNA on the hair. But there really is. There's actual residual crumbled up DNA on your hair shaft, and because they have so refined the way that they test the hair, that they can now say that it's, and again, they don't say 100%, it's a concordance. Yes?
Yeah. Talking about familial DNA, maybe I had it wrong. Did the courts not recently deny the use of familial DNA in court trials now?
I don't know. I've never heard that. I don't see why they would, because actually there's a case where they found out who the Zodiac killer was in California, and the way they did that is, if you did Ancestry, you had to sign a release to have your DNA data put into a database.
Right.
You could deny it, but a lot of people just say, sure, why not? And what happens is, this is an enormous database now, plus the fact that they have a database. Think of all the databases they have. Everybody who goes into the armed services has to submit a DNA. Everybody who ever committed a violent crime. And there are various other reasons. People who decide to enter into a disease study. So they have all of this DNA, and so what they do is they'll take the evidence and because it's all computerized now, nobody sits there, does it by hand. It's computerized, and it'll come up with significant matches, and then they have to go carefully through it. And what they'll find, they won't find the person usually. They'll find a cousin, an uncle, a brother. They'll find a relative. And then they'll go and question the relative. Now, the Zodiac killer was dead. Okay, he had already died, but when they interviewed, I believe it was his nephew, everything fit. And so this was a case that was solved through those. So I don't know why they would ever not admit that into court.
No, it actually started ancestry that, the people were putting the information in, and then the courts, it was some trial or whatever, that the courts actually got a court order to take that information, to use it in the court trial, and Ancestry said they're not relinquishing that information anymore, and then went another step further, and I thought that it actually got denied.
It may have something to do with permission.
Yeah, I don't know if it's a permanent denial, or was that one case where they denied it, but I remember hearing something.
But I would imagine, because of the sensitivity of that information, that they will have to come up, again, just like the Daubert case, they're gonna have to come up with rules for this. And I would imagine that the case that they were using, that there was a question about permission given.
Yeah, I mean, that's it.
We'll see. Tune in. Okay, so what else have we got here? We're almost done. All right, so we did this. Okay. So he was never convicted of those 11 murders that were linked to him, but I believe he made a jailhouse confession too, but I just don't remember. I know he was murdered in jail. Okay. So again, this is what we were just talking about, how databases really are so helpful to law enforcement now. Some people will say it's an invasion of privacy, but I would say you have no privacy. If you have a cell phone, you lost your privacy some time ago, okay? And you can deny, you could say, I don't want you to use my DNA, so you have the ability to do that, and I think those, pursuant to what you were saying, I really think that's going to be clarified in time. Did you ever see the movie Gattaca? Yeah, this was a young man who didn't have the right DNA to get ahead in life. It's a dystopian future novel where they test your DNA to see whether you have the right stuff, right down to your DNA. Okay. Science provides the methods, and humans make the decision. Also, you ever hear of the Innocence Project? That was Barry Scheck, who was one of O.J.'s original lawyers, by the way, and Peter Neufeld. And they were working with students, law students in the Cardozo School at NYU. And the graduate students, the law students, were investigating cases where men were convicted of violent crime, murder, rape, whatever, and they maintained their innocence. They never pled out, they never admitted, they affirmed that they were innocent. And so what they did in this case was they raised the money to get the evidence, to have it tested, and lo and behold, some of them were innocent. And so there are people who are wrongly incarcerated, and there are a lot of people who aren't incarcerated at all, actually. But that's it, okay? So if you commit a violent crime, you go into CODIS. CODIS is the Combined DNA Index System. And that's the first thing that they look at when they have a crime, and they have, there was a series of rapes in the Upper West Side of New York, and they had a complete DNA profile on the person, and they were able to find him through another database, but not CODIS. So it's an important tool, but you're still not going to find everybody. You're still not going to be able to convict everybody. So that's it, folks. There's other ways.
That's all, folks.
I'd like to show you one other thing. Remember Butch Cassidy and the Sundance Kid? DNA was used to prove that, you remember the end of the movie, they were in Bolivia? And they were supposed to
They
kill
got shot
everybody. And so there were these two people who were really Butch Cassidy and the Sundance Kid fans, and so they came across this story that in this little town that was very close to where they supposedly were killed, there was a grave where a gringo was buried. So they got a guy by the name of Clyde Snow, who you see, well, that's not Clyde Snow, who is an anthropologist, to go down there and check it out, to get permission to disinter the body and see if it was Butch or Sundance. It wasn't Butch, it was Sundance. No, it was Butch, not Sundance, because one was tall and one was short. And they found this skeleton, and they measured the bones, and the telephones were too short for Sundance, so they pursued Bush. And they had this computer program which takes a photograph of you and the skull, and to see if that photograph that face could match up with that skull, and it did. And so then they went one step further and they went to relatives and they got a DNA sample. No, not a match. Wrong Ringo. But yeah, there's Clyde. Here he is. Here he is. He's dead now, unfortunately. And of course, it was great because they had photographs of Butch. If they didn't have photographs, this would have been much more difficult to do. So it goes on and on. That DNA is, I have another program where I use DNA to solve ancient mysteries instead of just murders. So it's such a fascinating subject. Okay, thank you very much.
Thank you.
Yeah.
Even with things like the Innocence Project and how DNA is usually what exonerates the incarcerated person. Where is the expense? Like, where's the biggest expense on when
Where's the biggest one?
Where's the expense? Like, what's the biggest?
Oh,
part?
they
Like,
raise
why does it
money.
take so long?
There are 503C1. They raise money.
Oh no, I know that, but like, if say I get put in jail and I say I didn't commit this crime, why does it take so long in order for these people to then have their
First of
DNA
all-
gone through?
First of all, most of these people are indigent or very poor. They can't afford the dream team. They get a public defender, okay? And they are processed through the court very quickly. I hate to say it, but if you don't have that money, you cannot pressure to have that evidence processed the way you want it to, so they end up in jail. These men in the Innocence Project were all in jail.
Oh yeah.
They were all in jail and convicted and whatnot, but they maintain their innocence. And so, but as I said before, they save the evidence forever.
Yeah.
Which is really good. So that's how that works. Anybody else? Okay. I hope I didn't frighten you.